right. Well, hello, everybody, and good morning or good afternoon or good evening, depending where you're joining us in the world today. I can see we have quite a big distribution across the globe today, all the way from the US to Japan, it looks like. So thank you all for being here, whatever time it is in your home country today. I myself am based in Atlanta, Georgia, and it is about 10 a.m. and it's a beautiful spring morning here. Everything is blooming. It's a very nice day outside. Well, thank you for joining us today, where we'll be talking about cell cycle analysis, broad overview of applications, the assay, and a little bit more focus on the analysis since we're discussing this in a context of a FlowJo webinar. But before we start, I always like to get a sense of not only where everybody is joining us from, but also everybody's kind of experience level with the topic. So before I navigate to the slides, I'd like if you could share, what your experience level is with today's topic. How would you rate your experience level with cell cycle analysis? Is this something you've never performed, but you're here to learn because it might be something you do in the future? Have you done some univariate cycle analysis, so a DNA stain like DAPI or PI? Or have you done more complex cell cycle analysis on what we would call two-dimensional or three-dimensional or multivariate cell cycle analysis, whereby you have not only the DNA dye, but also maybe an intercalating dye to assess DNA replication or other protein markers for different stages of mitosis. So I thought I would just take a moment to see, where everybody's experience level is, so I have a sense of where I should, spend more time on certain parts of the presentation. Okay, so yeah, it looks like we have definitely some univariate cell cycle analysis experience, so that's good. It's probably the most common application that I see myself. And it looks like we have somebody who's also done some more complex modeling as well. Okay. All right, great. Well, I'm going to go ahead and leave this open in the background in case anybody would still like to participate, but I'm going to navigate now to our slide deck for today. All right. Okay. So let's go ahead and get going. So again, today we're talking about cell cycle analysis in FlowJo version 10 software specifically. Not to say that some of these manual gating approaches couldn't be done in FlowJo version 11, which is our newest version. But the modeling and the plugin we have to automate some aspects of multivariate analysis are currently only available in version 10. So that's what I'll be giving my attention to today. So my name is Veronica Obregon-Perko. I am a senior scientific advisor with informatics. I'm offering support for FlowJo software, but also just general consultation for flow cytometry data analysis. Following today's session, if you have any questions about this or other topics, don't hesitate to reach out by email. Always happy to lend a consult through emails, or we can also set up a screen share if there's any kind of data that you would like to review to get some advice on strategies and best practices. So a couple of housekeeping things. In terms of format, I'm going to spend a little bit of time in a slide deck this morning just to review, high level, what is cell cycle analysis, what are best practices for the assay and also for the analysis portion. And then I'll spend the rest of the time in the software just demoing all of our modeling options in FlowJo Software version 10. The session is being recorded, so you'll be able to access this recording from our website, from our learning module portion of the website. We host live webinars, but they all get recorded and added to a library. So you'll be able to access this at a later time. If you have any questions at any point, you can of course email me and send them after, or you can place them in the chat. I'll do my best to keep an eye on it. You don't have to wait till the end. If you think of anything in the moment, feel free to post it there and I'll do my best to answer it. Okay. All right, so let's get started. So, quick disclaimer slide for those of you that follow BD Biosciences or FlowJo Software on LinkedIn or social media. You may have heard that BD Biosciences has been acquired by the Waters Corporation. So, we'll be collectively referred to now as Waters Biosciences. No action needed on your part, right? This doesn't change anything about your FlowJo licensing or licensing structure, access, things like that. But we're just passing along the information because you may start to see some rebranding of materials or social media pages as time passes. So we just want to be transparent with our customers. Okay. So what are the learning objectives for today? So we'll have a short introduction to the cell cycle, how it relates to cell cycle analysis. What are the different types of assays? And it's important to establish those because they change how we analyze the data depending how many variables have been added to the model. I'm not going to spend a lot of time on assay optimization. That's really an entire topic in itself, but I thought I would spend just a few minutes mentioning some of the pitfalls we see that often hinder an interpretable analysis. So I'll have just a few suggestions for you for when you're optimizing your assay, but I'll direct you to some of my favorite learning resources that dig into these topics in a little bit more depth and really get into the details of dye selection, staining, time, lengths, things of that nature. Okay. And then lastly, we'll spend a little bit of time in the software just going through basic operation of these tools. So what is the cell cycle, right? So cell cycle refers to stages of growth and division by cells, right? I think when we were all taking, high school or college level biology, we studied, mitosis and meiosis, using things like chopped up onion root and things like that. I can remember still, looking in the microscope and looking at different stages of mitosis, so different stages of cell division. And so the cell cycle does include mitosis, right? So cell division. But that's actually kind of the latter stage of the cell cycle. The cell cycle also includes everything that the cells must prepare for in order to not only grow, but replicate their DNA, right? So before a cell can divide, it has to prepare to split its content. And that also involves, doubling its nuclear content. And so in that regard, this all happens during what we call interphase. So during interphase, we have the kind of resting G0 phase, which goes into the growth 1 or G1 stage, which is where we are preparing for DNA synthesis. So mitosis has been complete. And now for the next stage, we're preparing to enter the S phase, which is where the DNA actively replicates, doubling itself to prepare for the division. And then there's the G2 phase where DNA synthesis has stopped. And now the cells are kind of expanding, right, preparing to enter the cell division portion of the cell cycle. So why would a researcher be interested in studying the cell cycle? Why would we need to understand the distribution of cells across the different parts of the cell cycle, whether they're in mitosis or in different parts of the interphase DNA synthesis, G1, G2? So these are a few just kind of broad applications for the technique, right? So in cancer, they can play a role in diagnostics and understanding if there are aberrant cell growths, and at which point do we start to see an arrest of the cell cycle, or in terms of kinetics of the cell cycle, how long is it taking for a cell to double in time? Those can be informative for diagnostics in the cancer field, and also the impact of therapies that will target rapidly dividing cells. So if we do have therapies that can target cancer cells, there might be certain stages of the cell cycle that will be a good target for disruption. And so it'll be important to know which of those stages are being disrupted by a therapy or could be disrupted by a therapy. In immunology, it can be helpful for understanding proliferative responses, either to an infection or some kind of a stimulus, maybe like a vaccine or a therapeutic agent. So it has applications in that arena as well. And also in pharmacology, drug screening, right, similar to cancer, understanding toxicity, right? Are there stages of the cell cycle that get disrupted or is equilibrium kind of disturbed in the presence of some kind of an environmental toxin or a drug that's being screened as a candidate for some kind of a therapy. Okay? So lots of reasons one might need to study, right, the cell cycle. So in terms of how the analysis is done, right, how do we understand at what point in the cell cycle a cell is in, and how do we know when cells have accumulated in any particular stage of the cell cycle? So the distribution of cells that have entered the initial and final stages of the cell cycle can typically be evaluated with a DNA stain. Okay, so at its basic level for cell cycle analysis, most assays include a DNA stain, right? And this is going to assess the nuclear content of the cells, right? The idea being that, you have a given amount of nucleic acid when the cell is resting. And once the cell has completed the S phase and has completed, doubling its DNA content, then we should see a roughly twofold increase in that DNA content, right? And you can see that as an example here, where we have MFI or so of about 100 for this particular DAPI stain, which targets the DNA. And so we can see that in G0, G1, it's at about 100. And then after the DNA leaves the synthesis stage, right, the cell has left the cell synthesis stage, or sorry, the DNA synthesis stage, and enters the G2, then we can see that that DNA content has doubled, right? And so now that MFI has doubled, gone from 100 to 200. Okay? And so we'll talk about how these approaches can be complemented with other variables as well. But at its most basic level, this is essentially how it works. Okay? And so I put this figure here just as an example to show the impact of different, agents, right, different drugs, and how they impact different stages of the cell cycle, right? So now that we have a clear picture of what the G0, G1 phase is and the G2M phase, which is the latter stage of the interphase, then we can see that there can be accumulations at certain stages in the presence or absence of different drugs, right? So we can see that with this drug here, we have an accumulation into the G2M phase, right? Very distinct from the control sample. And we also have other drugs that disrupt maybe staying in the synthesis phase, things of that nature. So in having kind of a control sample, we can assess disruptions in the presence of other, inhibiting or interfering agents. Okay. And so when we include additional reagents, we can get more information or more dimensions about the other stages, right? So when we have just a DNA stain, that's referred to as a one-dimensional analysis because we can really only confidently go off of the DNA content and assess whether something is in the G0, G1 or in the G2M phase. And all the other stages are kind of inferred or a little bit more nebulous. But if we add reagents that specifically measure those phases, we can be a little more concrete about it. So let's talk about some of the different assays, right? So we've been talking a lot about the one-dimensional cell cycle analysis, which from the poll I showed just a bit ago, it seems like this is the format most are familiar with. But just to set the stage, right, with one-dimensional cell cycle analysis, or what's also referred to as univariate analysis, so only a single variable, that typically includes a DNA dye, right? There's lots of examples. These are just two of the most common ones I see, which include DAPI and PI. And so essentially what happens here is that we're identifying the G0, G1 peak, and then we assume the G2M has about twice the DNA content of G0, G1. Okay? And so I know this axis is a little grainy, but I showed it in the previous example where we basically establish a discernible G0, G1 peak. We look for a roughly twofold change in MFI, and then that discernible peak will be assumed to be the G2M peak. Okay? And basically everything that is not falling in the distribution of G0, G1, or G2M is kind of assumed to be a part of the S phase. Okay, so the S phase is kind of inferred. We're not really directly measuring if a cell is actively replicating its DNA, right? It's more so just, well, if it's not in one of the other two peaks, it's probably in some intermediate phase here. Okay. So we can, of course, use a manual approach to analyze this data. I could draw range gates on each of these portions, maybe utilize FMOs or different drug controls to help me. But usually the preferred method is going to be to use algorithms to fit a model to the data. And that way we remove any subjectivity that might be in place as we start to analyze the different samples across different treatments or different time points for like a longitudinal study. Now, in terms of algorithms, there are two that are available in the cell cycle model platform within FlowJo 10. There might be others in the literature, other variations of these two models here, but these are the two that we have integrated, so those are the ones I'll discuss on the next slide. Okay. So one option for algorithmic modeling of univariate cell cycle analysis would be the Watson pragmatic model. Okay. So here's essentially what it's doing. It first starts by finding the G1 peak, right? So it looks for, a local maxima, probably on the lower end of fluorescence intensity. And then it does a curve fit for, Gaussian distribution, right? Assuming normal distribution of the data. Then it looks for the G2M peak, right? Essentially by roughly doubling the content of DNA in the G1 peak. Okay, so if it identifies the G1, it starts looking at roughly twofold higher for the G2 peak. And then once again has the curve fit to fit that data to that Gaussian distribution. Now, the S-phase identification is where this model differs from the other one we'll talk about, right? So that usually is the difference in these two, is how they identify or infer what cells are in the S-phase. So for the Watson pragmatic, it's using a probability-based piecewise integration function. So basically assuming the probability of membership varies linearly with DNA content. Okay, so it's looking at the DNA content within the presumed S phase. And it's a linear relationship where, if it has so much DNA content, it's assumed to be somewhere in this portion of the S phase. Okay. So that's what the model is doing there in the univariate. Now there's another option available in the cell cycle model platform within FlowJo 10, and that's the Dean-Jed-Fox. Okay. So the G1 peak and the G2 peak are basically found in the same manner, right? We look for the local maxima and we do the Gaussian distribution. But now it's a little bit different for the S phase. Okay, so here we're assuming the Gaussian distribution is kind of riding on a polynomial instead of in a linear fit. And that just gives it an opportunity to maybe handle a more complex data, right? If we have data within the S phase that maybe is not going to model well to a linear relationship, then maybe the Dean-Jed-Fox gives us an opportunity to, capture something that's not going to be captured well with the other model. Okay. And so it assumes the width of the S phase is a function of the G1 or G2 widths. And it also has an option to model a synchronous S phase, right? So if your model perhaps fits in an assumption where we think all of the cells enter the S phase at the same time and they leave it at the same time, there's a checkbox there where we can enable that assumption and try to fit the model a little bit better. Okay? Otherwise, it's assumed to be asynchronous, right? Cells are just entering the S phase at random points in time. Okay. Right. So that's one-dimensional cell cycle analysis, but if we want to get more complex with our approach, right, we can also add one extra variable and refer to it as a 2D cell cycle analysis. Typically, when people refer to a two-dimensional cell cycle analysis, they're referring to the inclusion of a DNA dye, which is largely mandated in all of these approaches. And then we add on a nucleoside analog, right? Common ones will be like BRDU or EDU. And so what's the difference here? Well, here you can see that we've got another moving part, right? In our previous analysis, we looked at everything as a histogram because we were only looking at one variable. But now we get an extra variable. So we turn our analysis into a biaxial plot where we look at our DNA stain versus our nucleoside analog, which is being detected with an antibody against that molecule. And so what that buys us is that we have an opportunity now to not only identify the G0, G1, or G2M phases with the DNA content like we've been doing before. So here's the DNA content, MFI of, maybe looks like maybe 400 or 500, something like that. And then it doubles to reach closer to 1,000 for the G2M phase. And with the univariate analysis, we would just say, well, everything in between those two peaks is going to be S phase assumed. But now we have the inclusion of this BRDU or EDU, which essentially is a nucleoside analog that gets incorporated into the DNA as it is actively replicating, right? So as that DNA is being synthesized, it is grabbing these kind of nucleoside analogs or mimics instead of a normal AT or C or G. It's grabbing these molecules, incorporating them into the newly synthesized strand, and then we can detect those molecules with primary and secondary antibodies. And so in that way, we can be a little bit more concrete about whether cells are actually in the S phase because they'll be positive for this analog. Okay. So distinct from one-dimensional analysis in that the S phase is no longer nebulous or inferred, but we can actually be more certain of cells that are in that phase. So in terms of an analysis, that at this point it's largely manual. You could just identify each of the phases based on whether they are expressing the antibody for the BRDU or EDU, and then also the amount of their DNA content. Okay, so using them in concert, then we get a sense of those three phases. Okay. Now we can get more complex, right? So my complexity will stop here at 3D cell cycle analysis, but of course there can be multivariate analysis that goes up to, 4D or even have a temporal component. But one of the more commonly used approaches is this 3D model here, where we're starting to build on the univariate and the 2D. Excuse me. And so in this model, we have still the DNA dye. We have still the nucleoside analog, but now we add on some kind of a protein. Right here, I listed the histone 3 because it's a popular example, but there can be other examples as well, like Ki67 or cyclin, depending which phase of the cell cycle you have an interest in understanding or measuring. Okay. So walking through this, right, we'll already recognize the plot on the left from our discussion on 2D analysis. Okay, so as per usual, we identify the G0, G1, and the G2 phases based on DNA content. Okay, so if we double the DNA content, we're going from the G0, G1 to G2. The S phase is determined by inclusion of the nucleoside analog, like EDU in this example. So here I can say G0, G1, G2, and the S phase. Okay. Now with the 2D cell cycle analysis, you might have noticed, let me go back a slide here, that the G2 and M phase are always kind of referred to collectively, right? Because we can't actually distinguish based on DNA content alone if a cell has left the G2 phase and began to enter the stage of mitosis, right? So they're always just kind of referred to collectively. But with 3D cell cycle analysis, right, if we include something like something to measure, histones that show up during mitosis, we actually have an opportunity to decouple those, right? So instead of just referring to this population as G2/M, we can measure the presence of this phosphorylated H3, right, which shows up in the process of mitosis. And when we have a population positive for that phosphorylated marker, then we know those are cells that have entered the mitotic stage of the cell cycle. Okay. So by including that third variable, we're able to decouple and be more discrete about being able to identify those different stages. And again, there can be variations on this assay. Some will use, for instance, like Ki67, which helps us, identify cells that are more in the resting phase, or different cyclin combinations that tell us more specific phases of the mitotic cycle. Okay. But this is just one of the more popular ones I've seen in the literature, so thought it was worth discussing. All right. Now with 2D cell cycle analysis and 3D cell cycle analysis, those approaches, until recently are mostly subjected to a manual analysis, right? We draw gates, we use controls to help us establish cutoffs for, what's a G1, what's G2, and what's positive for a mitotic protein marker, okay? But understanding that there's always an interest in trying to automate aspects of that analysis or at least get some help from algorithms. The FlowJo team developed a plugin that will help you automatically identify populations in different cell cycle phases. So for a long time now, we've had the cell cycle modeling platform in version 10, which we already talked about in the univariate portion of this talk, which just looks at DNA content models G1, S, and G2 phases, right? But we didn't really have any modeling for more complex analyses. So here, insert the cell cycle plugin, which has been created for two-dimensional and three-dimensional assays. Okay, so inclusion of a nucleoside analog or nucleoside analog plus some kind of a protein. Okay. So high level, the algorithm identifies high density populations. It does this with a tool called HDB scan. And then after that, classifies them based on the relative positioning to one another. Okay, so looks for these high density areas, and essentially, the distance between events in the visual space here, and using that distance, kind of classifies whether a cell is in the S phase or in the G2, etc. Now, just a technical note here that the tool is only compatible with Windows and ARM Mac operating systems. So if you do have more of an Intel-based Mac, I don't believe the tool will work for you. But if you have access to a computer with Windows or ARM Mac chip, you could run the analysis there, and then once the results are complete, you can take it back to your Intel-based Mac device if you do have that. But it's a good starting point. We'll go through the tool. I'll show you where you can get it and test it out on your data and share your feedback. All right. So again, I don't want to spend too much time on this topic, right? This is more of an analysis webinar, but oftentimes when I am consulting with users on their cell cycle analysis, if we start to have issues with resolution or the modeling isn't going well, typically that comes back to experimental design, right? So I thought it was worth mentioning some of the key pitfalls that I see with users or when I'm watching webinars and the speaker, mentions things that can go wrong in the assay. So I'll direct you to more learning resources on this topic, but just a couple of, high level points. Of course, we want to try to choose dyes that are compatible with flow cytometry. So if you do see any microscopy applications for cell cycle analysis, and you just order all those reagents and try to run it on a flow cytometer, it's not always going to transfer, right? The laser excitations can be different on a flow cytometer, the filter setups can be different, and also just the sensitivity, right? How bright something is. Something might look very nice and bright on a microscope, but once we bring it to the flow cytometer, we can't see it very well. So just make sure you are using dyes or dyes conjugated to antibodies for your proteins that are rated for flow cytometry and are compatible with the configuration of your cytometer. We want to aim for as bright a signal as possible. We don't, of course, want to oversaturate the system, right, where events are collected off scale, but we're really banking on that twofold difference between the G01 and the G2M phase. So if we don't get a very bright signal, that's going to be much harder to resolve. And especially for the DNA dye, it's important to do a titration experiment, right? We want to achieve a happy medium where we get a nice discernible signal between the G1 and the G2, but we don't want to increase spread of the signal, right? We want nice single peaks. But more importantly, we also need to make sure that every single cell has uptaken that dye, right? So if we don't saturate the system, the cells with more DNA are going to kind of hog all of the stain, and that leaves less for the lower DNA containing cells to engage, right? And so then we'll start to get a lot of variability in our samples or in our longitudinal study. So to get really good consistency in your cell cycle analysis for a longitudinal study or a multi-treatment study, where you're comparing different variables through cell cycle analysis, we really need to make sure the titration has been done appropriately. Okay, so don't overdo it, but we also need to make sure that every cell has an opportunity to engage that dye. And of course, in terms of not overdoing it, we don't want to put so much dye that we have introduced a level of toxicity to the cells. Okay, so we do still need to be gentle with the process and assess if we're killing our cells in the process of having them uptake the DNA dye. And just a note about fixatives, there's kind of two classes of fixation for cell cycle assays, alcohol and formaldehyde. I think alcohol is kind of the gold standard because it reduces, the width of our histogram peaks. It makes them a little bit cleaner, tighter, and so easier to discern a good G0, G1, G2M. But that being said, it's incompatible with a lot of fluorescent dyes. So if you do add a third component to your analysis, like the 3D analysis or other surface markers, we can't usually fix with alcohol. So then users have to switch to formaldehyde. But formaldehyde has its own caveats as well. The cells aren't going to be as stable as long, so you'll need to make sure you acquire them a bit sooner than you might be used to with alcohol, and also take care with fixation times, right? If we fix cells for too long in formaldehyde without washing it off, we can sometimes affect scatter properties. Okay, so just make sure you're following an appropriate protocol for your application. And be mindful of, when you complete the experiment and when you collect the data, so we don't have samples sitting for too long. Okay. Okay, and then so that's more on like the sample prep side. On the acquisition and analysis side, a couple more pieces of advice. So the main goal of cell cycle analysis is to aim for these low CVs, right? These are coefficients of variation, right? So when your peak is not so narrow and it starts to get wide and we have a lot of spread in the data, that really impedes the mathematical modeling and also your interpretation to establish what's G0, G1, what's G2M. So ways we can achieve that or help that is to acquire on a low flow rate. That's one piece of advice. So here's this graphic I grabbed from an open flow cytometry YouTube video, essentially showing that if we have a low flow rate, right, we have a better chance of cells passing through the interrogation point nice and slowly. We have less chance of doublet events, clumps of cells, and worse off, those cells being interpreted as a single cell instead of a clump of cells. And so we can get nice, cleaner, reliable measurements if we're just a little bit patient and run our samples on a lower flow rate. Now you can see if we get impatient, over dilute our sample, or we have too many cells and we're trying to acquire them quickly, we turn up the flow rate, and it kind of just becomes a crowded highway, right? Cells are bumping into each other, passing through the interrogation point. Um, at the same time. And you can see the impact of the data, right? How those CVs are just expanding. And I don't have as clear of an idea now of where my discernible G0, G1, and G2 peaks are. Okay. So it really can make a difference. So just try to be patient. You can always concentrate the sample, and that way you keep it on a lower flow rate. You should acquire and analyze on a linear scale. We are looking for just a twofold difference in DNA content, because if you put that on a log scale or by exponential scale, it's going to get compressed and hard to see. And so you might miss opportunities to optimize the voltage or optimize the titration if you're not looking at it in a linear scale. Okay. So I would say as soon as you're at the cytometer, and definitely when you're analyzing, it's going to be more appropriate to look at that data in a linear scale. Okay. Make sure you include cleanup gates and exclude debris in doublets. Okay, for cell cycle analysis, the exclusion of doublets is very important because doublets could also be interpreted as cells with a doubled DNA content, right? So it's not just a single cell with a twofold DNA content. It's twofold DNA content because it's two cells stuck together. Okay, so we need to be able to know the difference. And so having these cleanup gates to get rid of two cells that might have passed through the interrogation point at the same time is of critical importance. And, not a requirement, but it can be really helpful to try to have controls. Okay, so if you have cells that cycle through the cell cycle very regularly, or treated with some kind of an agent that stimulates progression through the cell cycle, or if you have different drugs that can arrest different phases of the cell cycle, these can all be very helpful for you as kind of like negative or positive controls, or sham controls, so that you can establish that model. If we're trying kind of a novel experiment for the first time and we don't know how it's going to affect the cell cycle, it can be a little nebulous to know what you're looking for. But if you have extreme cases of arrest or cycling, it can make it a little bit easier. All right. So, as promised, I'll direct you here to some additional learning resources on these topics. We're going to move on to the software now and talk about the analysis component. But I think it's worth mentioning there's a lot of information out there on the web if you are looking to build up your cell cycle analysis assay in your laboratory or optimize an otherwise problematic assay. One of my favorite resources was from the OpenFlow YouTube series. This is made by world experts in cytometry and also cell cycle analysis specifically. So they have two modules that discuss very thoroughly sample prep, assay optimization, and even acquisition at the cytometer. So I'd highly recommend those. I also found some kind of short PDF guides that were published by these two universities. So I'd recommend those as well if you would just like a little cheat sheet to keep at your desk. If you're looking for more standardized formal learning, ISAC through Cyto University actually has a course that you can pay to take, or I believe it's free if you have a certain membership level, but has various modules, and you can actually earn CMLE credit if you are looking towards getting a cytometrist certification. And methods and protocols, very world famous textbooks for publishing standardized approaches from world experts, and they do actually have one on cell cycle. So if you're more of a textbook or ebook person, that would be a very comprehensive one, and I think it even establishes protocols for different model species and organisms. All right, so I'm going to go ahead and move on to the demo. I'm going to turn off my video just so I give myself a little more room on the screen. And while I'm shifting gears, I'll pause and see if there's any questions as well. Okay. Well, seeing none in the chat, I'm going to go ahead and move on. But of course, if you think of anything, just go ahead and drop it in the chat. I'll do my best to answer. Okay, so I'm going to start with a univariate analysis first. Okay, so this is a one-dimensional experiment where we pretty much only have a DNA dye to use in the modeling. There's a time series here where the cells were stained, after certain time periods, so we can see if there's any fluctuations through the cell cycle. Okay. Now, before beginning, as we mentioned, it's going to be very important to do some cleanup gating. So I thought I'd show you, what kind of cleanup gating I did on these files. Okay. Let me just go up a level here. Okay. So just to quickly go through cleanup, I mean, I would say at minimum, right, we do want to try to exclude debris and large particles and focus on our cells. Okay, so for something like that, you can use a forward scatter, side scatter combination. And we can see here that I've captured some cells, presuming everything that's very small or very large and complex is probably just debris. So at minimum, I would at least start here. Of course, doublet exclusion is also going to be very important. Okay? Unfortunately, in this experiment, they didn't acquire a ton of parameters, so I kind of just used what was available to work with. But just to give you an example of one, typically for doublet exclusion, we would do something like forward scatter area versus forward scatter height, or side scatter area versus side scatter height, or side scatter area versus side scatter width. There are lots of combinations of doublet exclusion gates. But one that was possible, at least in one of the samples where they collected these parameters, is to do doublet exclusion based on your DNA content dye, right? So you could say, like, well, I'm looking at my DNA content area, DNA content width, and we would want cells that have a proportional ratio, right? The idea being that cells that have a disproportional ratio are probably, two cells that were stuck together, so they took longer to go through the interrogation point or something like that. Okay. So you could include something like this. So if you had a PI stain, you could do PI area versus PI width. And this way you have an opportunity to exclude that. So here I can show you the impact of it. So let's say I go into this single cell gate, and let's pretend we were going to start our univariate analysis at this stage. So here I have the DAPI stain for the DNA content. If I start to move my gate up into this area of presumed doublets, right, we can see how that increases very dramatically the amount of cells that are estimated to be in that G2 phase. right? If I bring it back down, we have presumably G0, G1, presumably S phase and G2M. But I get a very different picture if I include the cells that are off this kind of singlet ratio. Right now, I'm getting a false estimation of cells that have entered the G2M, but really those cells just have double the DNA content because it's two cells stuck together. Okay, so this is just to show you the importance of cleanup gates, particularly doublet exclusion. Okay, so if you didn't collect area and width or area and height in your DNA content die, that's okay, right? You can do it moving forward. But otherwise, you could just use your scatter area and height or area and width and just be as comprehensive as possible with that exclusion. Let's see. And I think I see something in the chat here. Um, Someone says, how would you gate on single cells and exclude doublets if you have polyploid cells? Oh, that's a good question. So yeah, polyploid cells can introduce a different level of complexity. I don't think I have any data to really show that. But I'll see if I can dig up some literature and maybe we can include it in follow-up emails. My guess is that we would start to see some differences in the DNA content, right? So instead of a 2N here, right, we'd see maybe a third peak of a 3N, 4N, 16N, so on and so forth. So, polyploidy can start to get tricky. I would imagine your cleanup gates might be similar because when we're looking at things like area width, area height, right, those have to do with, how long it took a cell to pass through an interrogation point and not necessarily how much DNA content it has. So I would imagine we'd still want to capture cells with a proportional ratio for that because here we're basically saying it's okay if the DNA content goes up, but that doesn't mean the cell should take longer to pass through the interrogation point, right? So I imagine your cleanup gates, like what you're seeing, the two on the left here, I think those are going to be largely the same, right? Maybe you'll have some larger complex cells off the forward scatter. So maybe your forward scatter gate might need to be a little bit bigger. But I do think the doublet exclusion is probably going to be largely the same. But unfortunately, I don't have any data to really show that right now. And, doublet exclusion can seem like a complex topic. I would encourage you to read a little bit more for everybody joining today about what these different parameters mean, right? Area, width, height, why do we use them? Why do we care? Because they can tell us a lot about, assumptions we make about the data in our analysis, okay? So actually, I'm going to see if I can pull it up real quick. I did have one resource that I always like using. Let's see if I have it open. I was hoping I did. It's one of my favorite resources for doublet exclusion. Maybe I can find it in my browser history. There we go. Yeah, I'll leave this pulled up for a second for anybody that wants to take a quick screenshot of it or something. But this University of Wisconsin flow cytometry laboratory, they publish a lot of these job aids about different flow cytometry topics. And I think this is one of the nicest breakdowns of doublet discrimination I've come across, right? So this just tells you the idea of, why we use certain combinations like width and height. And it's basically to exclude this possibility, right? Like ideally, I want to only analyze cells that are in a single, you know, uh, interrogation. And when we have cells pass together at the same time, right, it gives a false negative or false positive signal. Okay. So when we use things like width and area and height, right, we're assuming that things will have a proportional signal. Like it's okay if a cell gets brighter, but if it gets brighter and takes longer to pass through the laser, then that falls into the assumption that that's actually just two or three cells stuck together. Okay. So I would recommend this for a read if you find the doublet exclusion to be kind of confusing. But having, understood what those are, right, you can see why it's still important, whether it's a multiploid or single ploid experiment, that we would likely still need to capture things on the ratio. Okay. And what you'll probably see in a polyploid experiment is instead of just two distributions here, you'll have maybe one, two, three, four, right? So more distributions, but still nothing up with a longer kind of time of flight. Okay. All right. So again, just explaining the importance of cleanup here. Unfortunately, these parameters were not collected on the rest of the data, so I can't show it to you on all of them, but it was collected on one, so I thought I would just show it as an example. Okay. And the other thing I'll call out is scaling. Okay, so remember your DNA dye is going to be best on the linear scale because we're going to be looking for twofold differences here. Okay, so if I change this axis to, for instance, a log scale, right, that's going to make it a little harder for you as a researcher to find that intermediate point, but also for the modeling to do it as well. Okay. So you're going to want to stick to linear for that. If you have a lot of wasted white space here and you feel like you can't confidently see the S phase, hit the transform button, go to customize axis, and here you have an opportunity to kind of, remove or add range if you need it. And I will say this will dramatically help your modeling as well. Okay, because if you feed a model, which we're going to go through in just a second, really compressed data, it's just not going to fit very well, right? The model sees what you see. So take an opportunity to, adjust the min and the max so that we get a nice range of univariate values and we can clearly see the twofold change from the G0, G1 to the G2M. Okay. Okay, so all that being said, right, I did some cleanup gates on the rest of the sample, so let's go ahead and launch the cell cycle platform. So in FlowJo 10, you can access that in the Tools tab, and we have a biology platform, and you can go to cell cycle. Okay. All right, so let's go through this window a little bit. Okay. So here's the sample, right? There's the parameter in question. This is the DAPI DNA stain. Hey, the data, your data is in gray. Each of the components are shown with these shaded regions, so what the model identified as the G1 phase, the S phase, and the G2, and then the summary of the model, so the distribution is in that pink border line, okay? You'll get more options in these drop-down menus here. If the colors are, distracting for you or something like that, you can always use these graph toggles here. So if you want to turn off the model summary, or the components, you can do that. If you don't like the shading, right, you can do just solid lines, though it does get a little hard to tell when two lines are overlapping, but if you prefer that, you can do that as well. Okay, so a lot of stylistic things here. So what happens by default? Okay, so by default, when you launch the module, um, it'll be defaulted to the Watson pragmatic model. Okay, so that's the one that does more of the kind of linear probability for the S phase. Okay. The model will auto fit, right, when it is in the unconstrained version. Okay, so it'll be unconstrained to start, and it'll basically just follow those assumptions we talked about on the slide, right? Looks for the local maxima G1, looks for the local maxima G2, and then kind of infers the S phase. All of your stats are given here, so percent of cells in each of the phases, the mean of the peak for each of the phases, the coefficient of variation, which is kind of like the wideness, the fatness of the peak. And then the percent of cells that are below the G1 phase or beyond the G2 phase. Okay, so basically everything that didn't fit into the other components. Okay. So if everything looks good unconstrained, you don't have to change anything. Okay, if you're generally satisfied with the model, like, okay, yeah, I captured everything, I think that's good, you could just leave it as it is. Okay. So there's the Watson pragmatic. Let's look at the Dean Jet Fox, for instance, just so you can see the difference. Okay. Now, one thing you'll notice is not only that it's different, but what you want to keep your eye on when you're making adjustments is this RMSD value. That's basically how much does the model deviate from your data. So comparing the gray to the pink. And ideally, they'd be completely the same, right? There'd be no deviation in a perfect world, but this is imperfect, so all you can hope for is the lowest value possible. Okay. So with the Dean Jet Fox, I can see that the value actually went up to 4. Right? When I had the Watson Pragmatic, it was 3.4. Okay. So I'd argue that the Watson Pragmatic is probably doing a better job. Okay. Now with the Dean Jet Fox, you can also remember there's the assumption that all of my cells have entered the S phase in unison. So if I hit that, right, it really didn't give me much improvement. Okay. So I can see, at least in my case, I'm probably going to want to just go back to the first one. These are both published tools. I can't really say with certainty if one is stronger than the other. These are just models and they get trained on data. So your data has not been trained on these models. And so you'll just have to see what's going to describe your data the best. Okay. So at least in this data set, the Watson pragmatic is probably going to be what I stick with. Okay. Now, let's say I'm unhappy with the unconstrained, right? I feel like there's room for improvement. What are some ways we can start constraining the model, right? So here you can see everything is unconstrained. Okay, so I'm basically letting the model just do its best to fit the data. One thing I could try to do is say, well, the G1 CV and the G2 CV are not quite the same, right? Maybe I should assume they're supposed to be the same because the level of variation should be the same. So maybe I would say, actually, the G2 peak, I want to constrain that so it's the same as the G1, okay? And I can see that actually my RMSD went up a little bit. So it's probably not helping me a lot. So then maybe I'll turn it back to unconstrained. Right? I could do the opposite. Actually, I want the G1 to match the G2. Right? Come here, turn that. Okay, and that actually raised my number. So again, I'm kind of moving in the wrong direction at this point. So those are all possibilities. Okay. If you change it to N, right, you'll have to manually input these CV values. So, if you want to keep tinkering with that coefficient of variation, you can, right? It'll start at 1 and you can keep changing that value. And the value you're manipulating is here, right? So now it defaulted to 1. Before, I think it was closer to 9. So I start back there. I get closer to where I was before, right? Maybe I could say something like 10. Hit the enter button, right? It's going down a bit, okay? But at this point, it's not making a dramatic difference, okay? So I'll go back to just making it equal to the G2, or actually I'll go back to unconstrained and make this one equal to G1, okay? So at this point, what I'm constraining is the coefficient of variation between the G1 and the G2 phase, okay? Now, the other thing you could constrain is the mean of these peaks, right? It's going to be kind of modeled, but you could say, well, I want the mean to be based on a range that I establish. Okay, when you do that, that's going to require that you draw a gate for that, right? So if you come in and say, oh, I want this to be constrained here, this is my G1 range. I don't want it modeled. I want you to use what I've drawn in the gate, right? Then you can do something like that. I could do the same thing for the G2 if I'd like. Like, okay, this is what I think to be the G2. I'm going to constrain that. Okay. And again, my model, the RMSD is kind of flirting around 3.5. It hasn't changed a whole lot. Okay. But these are just things I'm trying to see if I can get that RMSD to come down. Okay. Now, you could also say something like, okay, well, maybe I want the CVs to both be unconstrained, but I want to kind of remove assumptions about the peak of the G1 and the G2. So I could say, well, the G1 peak, I'm going to restrain it to the gate I drew, but the G2 peak, I want it to be G1 times. So I want to be G1 times 2 is the default, because remember, G1 we usually assume twofold change for the G2. That's why it defaults to 2. But maybe in my model, it's not exactly 2, right? Maybe it's 1.95, right? And look at that. That brought my model down just a bit. Right now, we're back at 3.45, right? I could even maybe push the limits a bit to 1.9. Okay, maybe that overdid it. I'll go back to 1.95. Okay. Now, if you do the opposite, so if I go back to the range, and then I say, well, the G1 peak, I actually want it to be G2 times, right? In this case, 0.5 will be the default because your G1 peak should be half as much as your G2. Okay, so that's where these default numbers are coming from. Okay. But I think all in all, I was doing pretty good with the unconstrained. Okay, so I'm going to stick with that. And so, once you've achieved a model you're happy with, okay, the idea would be you should technically apply that settings, right, to the rest of the data. Okay, so if I do unconstrained, and I apply this model to the rest of the group, I'll apply it to the rest of my cleanup gates, then it's going to run an unconstrained modeling on the rest of the files. Okay, so if I go through the rest of the files, right, I can see in some cases it did okay, some cases less okay, right? So maybe I have a little bit more work to do on some of these. But it's a starting point. Okay, and then I can go to one, do modifications, and maybe reapply the model. Okay. All right, I think I see something in the chat. Let me take a peek. What is a suggested CV value to make sure the data is good? And if my cell cycle distribution is not as what the literature shows, is it okay? Edited cells have similar wild type. When do I do experiments? So this means my edited cells do not affect. Let's see. I might have to reread that. Let's see. So I'll take it one at a time. So what's the suggested CV value? There's not one I'm aware of. The lower it is, the better, because that just shows like the level of variability. So, if you can, ideally, of course, it'd be like 0 or 1, but I don't think we usually get that low. And so, if the number keeps increasing, right, that's kind of a sign that there's a lot of variability in your cells, which could be for biological reasons, but it could also be because of suboptimal titration. Okay? So, in terms of what's a good CV, I don't know if I have an answer for that. I don't know if there is one. I think what I would pay most attention to is stability within an experiment. So maybe you'll have some CV variance, but if your negative and positive peak, right? So if I go through my data, let's just ignore the model for now. Like my G1 peak is always around 40,000 MFI, right? Notice how it's always kind of in the same place. If within the same experiment, I'm seeing like major shifts in these peaks, like in one sample it's 40K and another sample it's 100K, right? That's going to be really hard to model because, I'm going to get a lot of fluctuation in those MFI values. I think standardization is important. Low CVs are important, but I don't know if there's any cutoffs for, a bad CV or a good CV. And then the follow-up question was, um, and if your cell cycle distribution isn't what the literature shows, is that okay? Depends on the biology, right? If you have a reason to justify, um, distributions in the equilibrium, then I suppose it could be valid. But if it's just a standard cell cycle assay on a well-established cell line and you're not seeing what you see in the literature, I'd start to think about my sample preparation and whether I've achieved saturation of that dye. Maybe my fixation is too long, it's not long enough. Maybe I'm running it on a high flow rate, right? Because these are all going to impact, right, the distribution of that dye, right? So I suppose it could be possible, but I would just, if I were reviewing a paper, I would just need a justification on why I'm not seeing what's been established, and also, be sure that my protocol is really optimized. Okay, and now you can include controls for things like that. So you could use your U2 OS cell line and try to include different pharmacology agents to arrest different phases, and that gives you a little bit more leverage for arguing why your data looks different. Okay. Okay, and then I see another question here about polyploidy, I think. These algorithms won't fit polyploidy cells. What would be the ideal approach for univariate cell cycle analysis for polyploid cells? Yeah, Hadika, that's a good question. So yes, we are aware that this model and many of the ones out there, if not all of them, deal with polyploidy cells. It's my guess that that is still something of a manual analysis approach, whereby you'd probably have to be looking at these peaks, and if you're doubling or tripling of the DNA content, those would just have to be established with range gates and things like that. So, I'm less familiar with polyploidy, but I imagine that it is just something of a manual process where you're looking for these. Oh, whoops, didn't mean to draw that. You're looking for these more intense DNA content peaks, and having to just manually capture those either in a histogram format, or if you're in a format like we were looking at earlier, something like this where I have like, 1N, 2N, 3N, and just kind of capture those with individual gates. Um, Okay, let's see. There's another question here. If the settings don't work for all samples, which do you use? So that's a good question, Manuela. So, everything always comes down to controls. So having controls can be helpful to at least give you a sense of what the model should look like. Even in this example, I can see that in some cases, like it's not doing great for some of these. So there's a couple of approaches, right? I could either, go back to the control, tinker with it a bit and see if I apply those settings globally, if I get any improvement in, say, situations like this, right? The other approach is to maybe I should have started with this sample, right? Like maybe this is the one where I should start building the model and applying it to the others, right? Maybe the one I started with wasn't actually representative of everything. So maybe I should start with something else. The other approach would be, perhaps it's okay to constrain the model in certain samples. So maybe these settings are okay for this sample, but when I come to this one, I might need to do some constraints. I think what's going to be harder to sell is changing models completely. Okay, so if I have some samples where I stick to the Watson pragmatic, and then some samples I decide to change to the Dean Jet Fox. Like those are going to be mathematically different and they make different assumptions. So I would say if you largely just stick to one and you do slight adjustments on a sample specific basis, it's probably okay. But in general, the settings should be largely the same across the samples. And sometimes that can just have to do with which sample we establish the training on. Okay. Okay, there's another question. Do we need to run the samples with water buffer before we use for cell suspension? I'm not sure I understand that one. Do we need to run the samples with water or buffer? Water buffer. I'm not sure, Haidao, if I understand your question. I don't know if you want to elaborate a bit. I'll also add that I'm not an expert in cell cycle assays, right? I've helped users with the analysis. The assays are not something I have a lot of direct experience with. But those webinars I directed everyone to at the end, I think might lend a little more support on that. But I'm also just a little unclear on what the question is. So if you want to elaborate, I can do my best. Okay, since we can see events in the water buffered. Um, I'm not sure, Haidao, if your question is if we need to acquire water on the machine before we acquire the samples. It's always a good idea to clean the machine before you acquire samples, so it wouldn't be a bad idea to just run water or buffer or PBS, whatever your SRL or core facility recommends for the cytometer. It wouldn't be a bad idea to run a little bit of a rinse or a cleanup before you put your sample on. Especially because DAPI stain can be very intense. So if the user before you didn't do a very good job of cleaning the machine, their DAPI could contaminate your experiment and give you false estimations of DAPI content. So it's never a bad idea, I think, to just do a little bit of a kind of a rinse wash before you put your sample on the SIP, if that's what you're asking. I think that's a good idea. All right. Great, you're welcome. Okay, so we're running short on time, so I'm going to go ahead and move on to the 2D and 3D cell mo models here, just so we can start to wrap up. So, just to show you, this is the cell cycle model in a nutshell. What can you do with these outputs? Oh, I forgot to. Let me bring it back. Okay. With the outputs, you got a couple of options. One is in this first drop down, if you would like the gates that represent these phases, you can hit this checkbox here and it will give you those gates in the workspace, right? Oh, whoops, what happened? There we go. So here's the three gates, right? G1, G2, S. You'll notice they'll have some overlap, right? Even when we look at the model, that's just because the cell is being estimated, right, for which phase it's in. Okay, so you might get some overlap if you do like an overlay in the layout editor. But you could also manually draw these gates if you want to have kind of mutually exclusive membership. Okay. All right. And then in the layout editor, a couple of options as well. If you open up a new layout, if you drag that model into the layout editor, right, you can get a summary of your analysis here. We can also batch this, right, so that I get that same report, right, for all the samples, right, and it gives me all the statistics of interest there. And if you go to the table editor, right next to the layout editor, I'm going to make a new table here. If you drag in the module node here, we'll give you a table with the stats, right, which you can also create, right? And so you can see here, I don't have any stats for one of the samples, but you can see all the same statistics that were in that module, you get them per sample, okay? And then you can export that table as per usual, okay? Right, so those are the outputs from the one-dimensional univariate analysis. So what about the 2D, 3D analysis? Okay, so I do have a data set I can show you that with. Let me just get that workspace opened. Okay, so this is a two-dimensional cell cycle analysis. Okay, so in this case, what are we working with? We're working with cells that have not only the DAPI content or DNA content dye, but also the nucleoside analog like EDU, so we can look for the synthesis stage. Okay? So again, just to show you my cleanup gates. Now in this case, these parameters were a little comprehensive in their collection. I'm going to open them up here. Okay. So again, debris, forward scatter, side scatter, traditional doublet exclusion with forward scatter area versus forward scatter height. And now in this case, these users did acquire area, height, and width, I believe, on their fluorescent parameters, including the DAPI stain, right? So I also did a nice doublets gate here to make sure we get rid of cells that are presumed doublets because their DNA content is higher, but like their time of flight isn't, or something like that. There weren't really many, right? Most of them were cleared with the previous gate, but could still be important to remove those. Okay. And then here's the kind of key plot that is of interest, right? So we're looking at DAPI versus the EDU based on DNA content alone, right? Around 2 million for the G1, closer to 4 for the G2M, and then we have the EDU stain for the synthesis. Okay. Now, normally you would just have to do manual gates here. Okay, so I could take like a rectangle gate and just kind of come in, right, and start identifying these things. So that would be the S phase, come in here, and this would be like the G0, G1, right, something like that. Oops. I don't know why this dash isn't working. Okay. Right, G0, G1, and then, same thing here. So I think you get the idea for anybody that has done a 2D cell cycle before. Okay, so you'd be kind of, restricted to this approach here. Now, you could do this for the rest of the samples, of course, but we also have a plugin if you do want to lend a little bit of automation. Okay, so where can you get this plugin? If you go to the FlowJo website, in the download section, there's the FlowJo plugin exchange. Okay, here, if you type in cell cycle, that'll be the fastest way to find it. And you'll see here a cell cycle plugin. Okay. A little bit of note about what it does, its compatibility, the authors who created it. So you'll download the plugin. Once you download it, it will come into your downloads section of your device. What we need inside that folder is this jar file .jar. The jar file will then need to go into a plugins folder, which you can create in your applications if you're a Mac user. If you're a PC user, we give you a plugins in your FlowJo program files, or you could just make one local on your device. Okay, we have videos on our website about how to install plugins, so I don't want to spend too much time on that. But once it's installed, right, it will be available for use here in FlowJo, right? We'll have to set the path in the preferences, and then you can go to Workspace plugins, and then there's the cell cycle plugin. So if I decide I don't want to do these manual gates, let me just clear these out of the way. Yes. Okay. I've decided I want to just use the modeling. Okay, I'll select the population I want to run the modeling on, go into the cell cycle plugin. Pretty simple interface here. It's going to ask you, well, what is your DNA stain? It will try to find it automatically, but if not, you can just help it along. Okay. And then what's your DNA stain? Same thing. If you do have 3D analysis, you can hit the checkbox and then just tell it what is the third dimension, right? In this case, it could be like a histone protein, something like that, okay? But we'll just do the 2D for the sake of the time here, okay? All right, so then I'll say okay. And it's going to take a minute to run because basically what's happening is this algorithm is looking at the density of this plot, right? Where are there regions where there's like a lot of red, yellow, green, right? Where there's a lot of cells in the same area of the plot. And then it focuses on those areas and then looks at the distance of events in those areas. And based on the distance, we'll say, well, these are really far from these, so they must be their own classification. These are really far from these and these, so they're its own. So it's just classifying the events based on where they're found in the plot. Okay, so kind of a probability of, where a cell should be based on its location. Okay. So once it's done, you're going to see three or four populations here, depending how many it finds, right? So we've got four. So it identifies the G1, the G2M, the outliers, and the S phase. Outliers are basically anything the algorithm couldn't classify based on density. Okay, so I'll take you to the layout editor to give you a stronger sense of what that means. Now, one thing to keep in mind with these is you're not going to get any gates, right? Notice there's no gates here, right? So if gates are important to you to visualize, then you'll need to stick with the manual approach, okay? But if all you care about is just classifying the cells automatically, then you are going to get those classifications, but you're not going to see the gates that gave rise to those, okay? So what I mean by that is this is what your gates look like, right? These are just classifications, whether a cell fell into this, or this. Okay? And if you have the M phase, you'll have another class here. Okay. So how did the classification do? Let's take a look. I'll go to the L-shaped layout editor here. Right, let me take that final plot we were looking at. Okay. And let's just do a simple overlay, right? Let's take all of these populations that were found, put it right on top, and get a sense of how it performed. Okay. So we can see the G1 in yellow, G2M in green, S phase in pink, and the outliers in blue. Okay, so notice how the blue was kind of, it's kind of sporadic, right? It tends to be, maybe I can move this layering a bit, or actually I'll get rid of this layer just to show you. Okay. Right, the blue tends to be kind of, in the outskirts of the S phase, right? So like these events, for instance, they're kind of in between the G1 and the S. So the algorithm's not sure if it should put it here or here. So it kind of just puts them there, right? As an outlier. Okay, so it does some binning decisions. But overall, it does a pretty good job. I'd say it's mostly accurate, but maybe some events kind of get left behind just because, this is a density-based tool. Now, could we improve the algorithm? Perhaps, right? So maybe if I got rid of some of this white space and enhance that separation a bit, then maybe I'd be able to classify these a little better. Maybe I could try a different sample. So there's ways we could try to improve it. But again, it's just a quick, classification system for you if all you're after is just lending some automation to your analysis. All right, I do see something in the chat. Let me pop over again. I tried this plugin for my 2D analysis. It didn't work and gave me an error to contact the technical team. Okay, Hadika, that's a good question. It's hard to say when I can't see, what the issue was. So if you want to send me an email, which I'm gonna pop up here in a moment, you can perhaps share the data and maybe a screenshot of your workspace setup, and we can run it on our end and see what the issue is. Okay. I will remind that the plugin is only compatible for ARM, Mac, and for Windows. So if you are on an Intel-based Mac, I don't believe the tool will work. Okay. All right, so that's pretty much it. And then I could take this node, drag and drop it to the rest of the group, which I won't do because it's going to take several minutes to calculate for everything. But it would be very similar to what we did for the 1D analysis. Okay. All right, so let's go ahead and start to wrap up because we are just past the hour and I want to make sure everybody has time to get to other commitments and things like that. Okay. Okay. So as we start to wrap up, something new we are trying on the informatics team is offering some virtual support. We always offer virtual support through email, of course. You can always reach out. But just to kind of give you, a sense of a deadline or a time frame, we're setting aside office hours to discuss specific webinar topics. So since we discussed cell cycle analysis today, I'm setting aside some office hours on March 18th where you can book some time with me to discuss cell cycle analysis, right? You can bring your data, questions about your experimental approach, and we can try to have a discussion about it. So if you scan this QR code, you should be taken to a booking page where you'll be able to schedule some time with me that day. Understanding that this is a global event, the time zones might not work for you. If you don't see anything on that page that works for you, then of course you can just send me an email and we can try to find a different time to meet. Okay, but that's just a starting point for the office hours, just so that we can present some immediate options for availability. Okay, so here's my email one last time. If you have any questions, you try the plugin or the built-in univariate model and you run into any technical issues, don't hesitate to reach out. Happy to help. Common question is if the session's being recorded, where the recording is going. I'll go ahead and take you back to the FlowJo website. The recording will be posted in the Learning tab, Webinars. This is where you see all of our upcoming webinars, but you can also see all of our previously recorded ones, okay? So in the next couple days, I'd say you'll be able to see the link to the recording here, okay? All right, that being said, I'll go ahead and put my email just one last time. I'll take a look at the chat to see if there's any questions. Otherwise, I will stop the recording shortly and wish you all a good rest of your day. Thank you so much for spending time with us and hanging in past the hour. I hope the session was informative. And if you have any questions moving forward, don't hesitate to reach out.